everyone, my name is Aurelia and my teammates are Shabrina, Nasywa, and Fatih. This video is created for our STEAM Fair project. Our project is to conduct an experiment on seed germination in order to know the conditions necessary for germination. The title of our project is Environmental Conditions that Affect Germination of Seeds. Next, we'll be showing you a tutorial of the experiment that we conduct for this year's STEM Fair. We will plant the seeds in 4 different conditions. The conditions are lacking oxygen, lacking water, lacking heat, and all condition present.
We're going to use 4 plastic cups, oil, 4 pieces of cotton, tap water, boiled water that was cooled, and the last 20 chili seeds. We use chili seeds for this experiment. Put 2 pieces of cotton into each plastic cups. Put 5 seeds into each cup and place them on top of the cotton pad.
Remember that we are going to make each cup as different conditions. For the first cup, the condition is lacking in oxygen.
Pour the boiled water that was cooled into the first cup. I'll just pour the water until it covers the entire cotton. Pour the oil into the first cup. The oil on top of the water will stop any extra oxygen from getting back into the seed. So those seeds should have water and suitable room temperature, but they would not have oxygen. Moving on to the second cup, we don't need to add anything including water, so let it be.
Next is the third cup. This cup will be the same as the usual plant that gets the right amount of warmth or heat, water, and oxygen, or you can say all conditions present.
We pour the tap water into the third cup with the same amounts as the first cup. The last cup, which is the fourth cup, will be lacking in heat. Pour the tap water into the fourth cup with the same amount as the first cup. Last step, put all the cups in a sunny window for a week except for the fourth cup. Put the fourth cup into the freezer for about a week.
This is the picture of the seeds on day 7 in cup 1. The seeds are planted in the absence of oxygen. On the last day of the experiment, still no signs of germination were present in this cup. The germination has stopped presumably in the absence of oxygen. This is the picture of the seeds on day 7 in cup 2. The seeds are planted in the absence of water. On the last day of the experiment, there were still no signs of germination in this cup, and still the same as the first day. The germination has stopped presumably in the absence of water. This is the picture of the seeds on day 7 in cup 3. The seeds are planted without the absence of any condition required for the seed to germinate. The germination has stopped presumably in the absence of heat or warmth. This is the picture of the seeds on day 7 in cup 4. The seeds are planted in the absence of warmth or heat. On the last day of the experiment, the true leaf have completely emerged. The germination process is complete.
Germination is a process controlled by enzymes in which the seed begins to develop into a new young plant. 3 main factors are needed for successful germination. The first one is water. Water allows the seed to swell up and all the chemicals reaction involved in the growth of the embryo to take place. The second one is oxygen. Oxygen is needed for aerobic respiration, which provides the energy which the embryo needs to carry out cell division and growth. The last one is warmth or heat. It is required for the enzymes to carry out respiration and cell division. Based on the experiment conducted by the observers conclude that the seeds planted without water, oxygen, and heat will not germinate. On the last day of the observers' experiment, the seeds planted by the observer in the absence of oxygen, water, warmth, or heat did not germinate. Meanwhile, the seeds planted without the absence of any condition required can germinate properly. In conclusion, water, oxygen, warmth, or heat are needed for seeds to germinate properly.
We already did the last objectives, which you can see at the beginning of the video until the conclusion of the experiment. So next, we will answer and explain the first objectives followed by the second objectives. Reproduction is one of the fundamental characteristics of all living things. Each kind of organism has its own particular method of reproducing. From the two categories of reproduction that I have mentioned, I will only explain sexual reproduction. Sexual reproduction is a type of reproduction that involves a complex life cycle. It is the process in which new organisms are created by combining the genetic information from two individuals of different sexes. The genetic information is carried on chromosomes within the nucleus of specialized sex cells called gametes. Eggs and sperm are examples of gametes. Two of these gametes join and their nuclei fuse together. This is called fertilization. The new cell formed by fertilization is called a zygote. The zygote divides again and eventually grows into a new organism. Peduncle. This is the stalk of the flower. Receptacle. It is that part of the flower to which the stalk is attached to. The main function of the calyx and its sepals is to protect the flower before it blossoms, or in the bud stage. Petals. They are often bright in color as their main function is to attract pollinators. Stamens. These are the male parts of a flower. They are structurally divided into two parts. Filamen, the part that is long and slender and attached to the anther to the flower. Anthers, it is the head of the stamen and is responsible for producing the pollen. Pistil. This forms the female parts of a flower. They are structurally divided into four parts. Style. It is a longer, slender stalk that holds the stigma. Stigma. The stigma contains a sticky substance whose job is to catch pollen grains. They are responsible to begin the process of fertilization. Ovary. They form the base of the pistil. The ovary holds the ovules. Ovules. These are the egg cells of a flower. Characteristics of insect-pollinated flowers. Large and brightly colored to attract insects, usually scented and with nectar. The pollen grains usually sticky or spiky to stick to insects well. The anthers are inside the flower. The stigma is inside the flower. A moderated quantity of pollen.
For the math part of our STEAM Fair project, there are two objectives. We will answer and explain the first objective followed by the second objective.
We have made 4 discrete data based on each condition. A simple introduction about discrete data. Discrete data can only take certain values. It is also based on counts. Only a finite number of values is possible, and the value cannot be subdivided meaningfully. For example, the number of students in a class. I will explain about mean, median, mode, and range. Mean is the average of a set numbers. The mean is the total of the numbers divided by how many numbers there are. Median is the middle number in a list of numbers sorted from lowest to highest. Mode is the value that appears most often in a set of data. Range is the difference between the highest and lowest value in a set of numbers. This is one of the discrete data tables that we have made. We choose only one table to explain, which is the all condition present table, because this condition is the only condition where the seeds are germinating. We pick only one seed from this condition to write on the table. On day 1 and day 2, the length of the shoot is 0. From day 3 until day 6, the length of the shoot keeps increasing. And on the last day of the experiment, the length of the shoot is 4.5 centimeters. To calculate the mean, add all of the values on the data and divide it by the number of values. And we have here the mean is 1.714 centimeters. To calculate the median, sort the values from lowest to highest and then find the middle value. And here the middle value is 1 centimeter. The mode from this set of data is 0 centimeter. To calculate the range, find the highest value and then subtract it by the lowest value. And we have here 4.5 centimeters for the range. A cumulative frequency distribution is the sum of the class and all classes below it in a frequency distribution. All that means is you are adding up a value and all of the values that came before it. Here's the cumulative frequency diagram that we have made. As you can see, some of the lines are mixed because most of the seeds have 0 centimeter development. I will explain the diagrams to make them clear. The pink lines represent the condition of lack of oxygen. The black lines represent the condition of lack of water. The orange line represents the condition of lack of heat. And last, the blue line represents all conditions present. For the pink, black, and orange line, it means that from day 1 until 7, the seeds did not germinate at all. So the cumulative length of shoots in centimeter from day 1 until 7 is 0. As for the blue line, it means that the seed is germinating well because each day it keeps getting longer and longer in centimeter.